Good morning, I'm Jessica Lovell. Today we bring you an encore presentation of one of our very favorite programs. We'll still be live with you this summer, Monday, Wednesdays, and Fridays, and then come September, we'll be back with our regularly scheduled programming. Until then, enjoy this encore presentation and make it a great day. The name of this show, of course, is Open Mics with Dr. Stites, but I am Jessica Lovell, filling in for Dr. Steve Stites this morning. And it is the first Wednesday of the month, which means it's time for Show Me the Science. Today, we are learning about what some doctors call a living drug, white blood cells, but genetically engineered to fight cancer. It's called CAR-T therapy, and today we're focused specifically on new research showing how CAR-T therapy can be more potent and useful. We'll meet a patient whose cancer is in remission after other treatments failed. And you'll hear why one doctor calls this the most exciting time in his career. But let's start with our morning rounds today. You may have seen headlines about a new CDC report warning of a deadly fungus that is showing up more and more in the United States. It's called Candida auris, or C. auris. Good news, there's never been a case reported in the state of Kansas, but hospitals and nursing homes need to be on the lookout. Joining us now is Maggie Rivas, an infection control and prevention nurse here at the health system. Maggie, good morning. How are you?
Good morning. Hi. Thanks for having me.
Thanks for being here. So healthy people aren't really at risk. This is not a threat to the general public, but why is it such a concern for healthcare facilities?
Yeah, you're absolutely right. The folks that are at the most risk for infection with C. auris are people who are admitted to an inpatient type care facility, whether it's a hospital or a long-term care facility or a nursing home setting. So the general public is not at risk for this type of an infection. But we're concerned about it because it is often resistant to many of the drugs that we use to treat fungal infections. It can be really challenging to identify in the laboratory, and it is capable of causing outbreaks in healthcare facilities, which is definitely concerning.
So the CDC used the word alarming when it described how fast C. auris was spreading. So how fast is it spreading?
Yeah, in 2009 was when it was first identified in the United States, and at first, over the first couple of years, it spread really slowly with only some cases in about 4 or 5 states. And the CDC released a updated report in the last couple of weeks, which is why it's been so popular in the headlines, noting that infections have increased dramatically over the last couple of years, with infections doubling from 2020 to 2021 and almost doubling again from 2021 to 2022.
So what is the health system doing to prepare for and prevent C. auris infections?
We are partnering really closely with our laboratory to make sure that we have the technology in place to identify this type of infection when we have a patient present to our health system. We're partnering really closely with our public health authorities, both local and state, to make sure we are informed of any concerns for cases or folks that are transferred here from other facilities from another state that might be infected with this type of organism. We've verified that all of the cleaning agents that we use are effective at disinfecting C. auris, and we're putting together an educational material that we would use to make sure that our nursing staff and our providers are really comfortable and informed when we do get a patient.
So are other healthcare facilities doing some sort of the same?
Yes, yeah, absolutely. And it's important for them to, like I said, with the laboratory, to make sure that they have the technology in place to identify it, because it can be tricky to differentiate C. auris from other types of fungal infections, and to make sure that they have their basic infection control practices in place so that they can really prevent that spread once it shows up at their place.
Maggie, thank you so much for sharing this info this morning. We appreciate it.
Thank you so much.
After chemotherapy and a stem cell transplant, the woman you're about to meet still had non-Hodgkin's lymphoma. That's when she heard about a study testing a new approach to treat her cancer.
My cards and I have a really good relationship. I'm going to do a magical star.
Okay.
So here we go. I do lean more towards the metaphysical and the power of attraction.
64-year-old Katie Kopp loves to help others heal and find their way.
To me, with the light, you're holding on to your heart.
Then she was diagnosed with non-Hodgkin's B-cell lymphoma.
When they told me, I was, okay, what's next? Let's get rid of it. I have things to do, you know?
But as we know, cancer isn't quite that simple.
I decided that I can heal myself. I'm not going to sit idly by and let the medical community heal me, which is really ironic because that's exactly what happened.
Meet Dr. Joseph McGurk.
Hello.
He is one of the world's leading experts in blood cancers and cellular therapies. Based at the University of Kansas Cancer Center, he works endlessly to put patients into remission.
It's all inspiring. It really is. I'm like a kid in a candy store.
A candy store filled with new therapies helping blood cancer patients live. Patients like Katie, who after failing several other therapies to treat her lymphoma, Dr. McGurk was able to enroll her into a new clinical trial. How new?
What does it feel like to be the first?
I mean, wow. Wow.
It's called Cas9 gene editing, but more simply referred to as CRISPR. And Katie is one of the very first in the world to successfully be treated with this new method. It allows scientists to precisely cut out any strand of donor DNA so it can be used by a patient.
Not only is it potentially a healthier, more effective cell, but it's something that we can access right away.
A type of off-the-shelf option for really sick patients.
We can make these CAR T cells for hundreds of patients from a single donor. We can freeze them, access them like a drug, where we call up and they FedEx them overnight, and we have them the next day to infuse into our patients.
And Katie hasn't slowed down. She's back to work at another one of her many jobs.
Go. All righty.
Oh, I've got a huge green light. You know, I'm like, yes. What do you want me to do? Yes. What am I doing next? Yes.
You're welcome.
It's phenomenal. And Jessica, truly, I didn't realize just how innovative it was.
If you told somebody just in the street, they would think that you were making up a tall story, but it's not a tall story at all. It's reality. It's extraordinary. It's gee whiz science, and I believe that.
In fact, CRISPR won these two scientists, Emmanuelle Charpentier and Jennifer Doudna, the 2020 Nobel Prize in Chemistry. Katie Kopp is living proof of their incredible discovery and Dr. McGurk's passion and execution.
I'm grateful for everything.
And we're so happy to have Katie Kopp joining us this morning. Katie, good to see you.
Thank you.
Good to have you. Also, that kid in the candy store is also with us. Dr. Joseph McGurk is a hematologic and medical oncologist and division director of hematologic malignancies and cellular therapeutics at the University of Kansas Cancer Center. So glad to see you two back together.
Great to see you, Jess.
Oh,
and
good.
Certainly great to see Katie.
I know, right here in the flesh, sitting at the desk with us.
That's right.
It's the best. We've been working on this story for several months, so it's just nice to have you. Dr. McGurk, before we dive into the research, let's take kind of a big picture look at this. And why is this kind of immunotherapy so exciting for doctors and especially for you?
You know, it's critically important to the potential cure of some blood cancers that historically were incurable, and we would lose patients to predictably almost 100% of the time. This is work that's based on the efforts of men and women in laboratories, as you mentioned already, too, across the world who have helped us understand how the immune system works, what goes wrong when it doesn't work, and then leveraging that knowledge to treat our patients. And so instead of chemotherapy, traditional chemotherapy, radiation therapy, surgical approaches, this allows us to take cells out of the patient's bloodstream historically, and modify these cells, specifically cells called T cells, engineer them genetically, as you described, expand them and infuse them back into patients. And using the patient's own cells, we have seen complete remissions in patients 50 to 80%, depending on the specific type of blood cancer. However, that's not good enough. We need to do much better than that and cure everybody. This needs to be effective in a much greater percentage of patients. And so one of the problems with using the patient's own cells is those T cells had let them down in the first place. T cells are supposed to fight not only infection, but also recognize and destroy any cells that have the ability to turn into a cancer. And that didn't happen in Katie's situation. Her T cells let her down. and the cancer figures out how to manipulate those T cells. In addition, Katie had a lot of chemotherapy before we saw her, and that beats up on not just the cancer, but the T cells as well. So this study that Katie participated in took a different angle and said, you know, let's use some young, healthy people who have never been diagnosed with cancer and have T cells that have never seen chemotherapy and therefore are more healthy and potentially more robust. There are major challenges. Those cells might attack Katie and cause a problem called graft-versus-host disease, or Katie's immune system might destroy them and reject them promptly so that they weren't effective. And so through this gene editing technology in our laboratories, we've been able to manipulate what we call third-party donor cells from healthy donors and infuse them into patients. And Katie is one of the first people on the planet to have received such therapy in a novel, cutting-edge research study that I'll be presenting the results of in Paris in just a few weeks at the Presidential Symposium. It's a landmark trial because we use somebody else's cells, their T cells, to treat effectively patients like Katie. And Katie's now approaching 3 years out from her therapy and remains in clinical complete remission.
And this is called the Carbon trial, and the key question of this trial was this certain kind of CAR T therapy called CTX110, finding out if it was safe and effective when fighting B-cell cancers. So we're going to talk about a little bit more even into what makes that therapy special, but and you touched on it a bit, but what makes Katie such a really, really good candidate for this? What made her so good?
Katie was a very determined, and still is a very determined young lady who, as she mentioned in the video, said, okay, let's get rid of this thing, I have things to do. And that has been her outlook and attitude the entire time through this therapy. She was a very willing participant, and we were in big trouble. Her disease was not responding appropriately to chemotherapy, and for those patients, only 7% will respond to subsequent chemotherapy. Only about 16% of those patients are alive at 2 years afterwards. We lose the overwhelming majority of patients in the first 2 years, most of them in the first year. So the odds were greatly stacked against us. We were trying to climb Mount Everest and getting rid of this rotten disease for Katie, and we hit a home run.
So, Katie, you're the queen of self-healing, and you hear about this clinical trial, and I know what you're going to tell me about how much you trust this man right here, but when you first heard about the option of a clinical trial, what were you told about it? What were your feelings about it?
Well, I was apprehensive, of course, at first because it was brand new, and the first thing I did was three-way with my sister and my niece and say, hey, this is what's going on, what do you think? And they're like, yeah. So as Dr. McCurk mentioned, I had been through quite a lot of chemo before then. So the option of possibly being in remission in a month, it was a no-brainer.
So tell me, though, Dr. Berger, what's a typical CAR T therapy? Like what's the process like if somebody were not to have this so-called off-the-shelf option,
yes,
as far as going on that search for a match?
Absolutely. So the Food and Drug Administration in 2017 approved the first CAR T cell therapy, and as you pointed out, this is living therapy. When we infuse it into patients, it doesn't go out of the system in 24 hours. We can detect these cells, these CAR T cells, for months, and in some patients even years afterwards. So if the cancer cells are still there hiding out and then they show themselves, the CAR T will go after it again. The current state with the FDA-approved constructs, and there are now 6 indications for CAR T cell therapy available for a variety of different types of blood cancers, is to take the patient themselves, collect, first get approval and work out the logistics, and time is not on our side. Mm-hmm. Patients like Katie, we don't have weeks and months, we have days to weeks to get this under control before they become too sick and are not eligible. So some patients come to us in the logistics of collecting their own cells mm-hmm. from their blood and taking those to the laboratory and spending the weeks necessary to engineer these T cells may result in a number of patients having progressive disease, and we lose them. They die before we can get them to the CAR T cell therapy. In addition, not everybody can have a CAR T made effectively. Most patients can, and we can engineer the cells, but some we fail, and in part that may be because we couldn't collect enough of their cells, their T cells, or their T cells were dysfunctional and just couldn't engineer them so that they would fight the cancer. When we look around our nation at the number of patients who might benefit by this therapy with lymphomas like Katie had, and other diseases like myeloma and other types of lymphomas and acute leukemia, what percentage of patients since FDA approval are able to access this therapy? 30% of the patients who should be eligible. So it's only a small fraction for a potentially curative therapy that has a remission rate in patients with acute leukemia of 80%, a specific type of acute leukemia, B-cell acute lymphoblastic leukemia. And in lymphomas, as I mentioned, response rates of 50 to 80%, complete remissions of 40 to 50%. And most of those patients who go into complete remission are long-term cures, more than 5 years of follow-up. And we have patients like Katie who are still in continuous complete remission. We consider that an operative cure for those patients. But many patients drop out. Not everybody responds. It takes us weeks to make the cells. Using a third-party donor, we can collect cells from a young, healthy college kid and make enough CAR T therapy for hundreds of patients, potentially, freeze it down in small aliquots in our laboratory, and then when the patient comes to us, those cells were made months ago, we're ready to go tomorrow.
Avoiding those patients getting sicker and sicker.
That's exactly right.
And this CTX110 is one of those off-the-shelf therapies, correct?
That's correct.
And you kind of touched on it, the Carbon trial results showed an overall response rate of 67% and a complete response rate of 41%. So what do we mean? What's the difference between overall response and complete response?
Absolutely. That's a very important question. Overall response means the tumor shrank. Complete response means we can't see the tumor anymore. And so overall response rate is a step in the direction of getting patients into complete remission. But the complete remission is what we're after, because that's what is potentially curative in our patients. Now, for many patients who have therapy like that which Katie received, this is a last stop gap measure for us to move forward with a potentially curative therapy. So those percentages are not acceptable either. And so we're working on the next generation of these CAR T cells to engineer them further so that they will be more effective in patients. This is just the beginning. And so this is an exciting signal. When you look at that and you say, well, Dr. McGurk, you said earlier using the patient's own cells, you got similar response rates. So what gives here? How is this an advance? Because we're using these third-party cells, it's just our first foray into this, the first generation. And again, there are men and women in laboratories, including in our own center, who are figuring out how to make these cells much more effective through further genetic engineering than the current constructs that we have. So this is an early signal that we can safely give cells from a third-party donor not related to the patient, that they won't be promptly rejected and they won't attack our patient and cause graft versus host disease. That's a huge milestone in the field. And so we will see this therapy expand in its application to many, many thousands of patients and to many different types of tumors, not just blood cancers, but solid tumors like breast cancer, colon cancer, lung cancer. And we have such clinical trials that are currently underway at the University of Kansas Cancer Center.
So what do you say to people who are skeptical about gene editing?
Healthy skepticism is important, and we don't know if these constructs are going to cause a problem 15 years down the road. So patients like Katie and other patients that we put on clinical trials have to be followed per the Food and Drug Administration guidance for 15 years afterwards to make sure that there aren't some late effects that these cells could potentially cause problems down the line that we don't know anything about today. But so far, the track record from the early part of this century in the last 15 years has been one of very, very good safety long term. There are toxicities associated with these therapies on the front end when patients receive them.
3 years out, Katie, any side effects during this time?
No.
Nothing. How are you feeling? Are you the perfect patient?
I don't know if I'm the perfect patient, but when I come in for my checkups with Dr. Shanae, she has claimed that I'm the perfect patient, which, I think they're the perfect doctors, so it's kind of a two-way thing. But
it takes two.
As far as my blood work,
doing good.
Yes.
And you're feeling
Yes.
good, though. Tell us about that. I mean, I know you're
feel
you,
really
and you have
good.
this super Energizer Bunny thing going on with you, but do you truly feel good day in and day out?
I do. It's a little annoying, I think, for some people, but I do feel good. I feel very blessed to be at the right place at the right time. And it's kind of ironic because the pandemic actually kind of helped, because I had I had the non-Hodgkin's and had a stem cell transplant, and that got rid of the non-Hodgkin's, but I still had the follicular. Well, then the pandemic happened. I didn't come back in for my checkup because I didn't want to hang out at a hospital. It blew back up into the non-Hodgkin's, which made me qualify for the CRISPR trial.
What do you make of that, Dr. McGurk? That's kind of unusual, just some
Yes,
strange star alignment.
Absolutely. Well, we saw a lot of this actually during the pandemic, that patients would put off their health care and their follow-ups because they were afraid to come to the hospital, because there are a lot of people who were admitted to our hospitals with
sicker?
COVID during that time, and they were afraid of acquiring this. And so we had patients like Katie who came to us with more advanced cancer than potentially would have been otherwise had we detected it earlier. And she had a large tumor in her abdomen, and so we had a lot of disease to deal with these CAR T-cell therapy. But the results have been actually quite remarkable.
And to be clear, we do not suggest people missing their appointments with you.
Absolutely not.
Okay, let's get with Dr. Dana Hawkinson. So, Doc, of course, we talk a lot about the COVID vaccine here on this program, but another example of amazing science. But there is a real concern of what's being called vaccine fatigue. Some people just getting tired of hearing about the vaccine. They could care less about the vaccine. A new study looked at vaccine fatigue. What do you make of that?
Yeah, I mean, I think this is a very real thing. We heard about this early on. Right now we are talking about COVID vaccines, and I think it's multifactorial. First of all, I think it does have to do with continuing to try and instill trust in those scientists, in your medical caregivers, but also understanding that in the big picture, overall vaccine fatigue. So we know that the science is changing, and as we have more information and more data, we are able to make different recommendations. And so I think it is very important to tailor messages about vaccines to specific populations. If we're talking about COVID vaccines, it's obviously tailoring those messages to those people that are unvaccinated, or tailoring the message to those people who are not up to date or fully vaccinated, especially if they are high risk. And then for vaccines in general, it's of course important to continue to endorse and tailor those messages to, say, parents to get their children vaccinated, to help prevent those children from being ill from those very easily prevented childhood illnesses, which historically have caused so much illness and complications for those children. So I think there are a multitude of factors involved in this vaccine fatigue for sure, but the messaging has to be multifactorial as well, and also, again, certainly focusing on certain groups. Hopefully we will get new recommendations for COVID boosters soon, and then again tailoring those to the most at-risk populations and tailoring them in a way that people can accept them, and of course continuing to endorse and instill trust in science and medicine as well.
Any reporters with us today? Let's get to our community. And our first question comes from Jeremy, and that's going to go to our kid in the candy store. Dr. McGurk, when you find out about a new game-changing treatment or technology like this, what is the first day like for you? He's imagining there's a lot of discussion and a lot of excitement, but when you really get that information that this is working, this is that gee whiz science that you always say, what's it feel like? What's that first day like for you?
It's loaded with anxiety and anticipation and hope. All rolled into one ball, and so everybody's the whole team, and most importantly the patient and their family, are waiting to see, is this going to be effective? Is it going to cause untoward toxicity? Is it going to get rid of this thing once and for all? And so it takes a brave patient who is determined to participate in such clinical trials. It's interesting. This is a great question because I was just thinking about this earlier this morning at University of Kansas Cancer Center. We're designing currently, in collaboration with the National Institute of Health and Children's Mercy Hospital, a study where we'll make CAR T cells at University of Kansas Cancer Center and send them to the National Institute of Health and Children's Mercy Hospital. These CAR T cells will be somewhat different than what Katie received. These CAR T cells, instead of recognizing one molecule, specific molecule on the surface of the cancer cell, where Katie's done beautifully, and many patients have, but other patients have relapsed, and one of the reasons is the cancer cells are smart and they pull that molecule off their surface, they can't be seen. So we're making a CAR T that will recognize 3 different molecules on the surface of the cancer cell. So when if we give it to our patient, if the cancer cell tries to outsmart us and takes one down, we still got 2 others that it's going to go after to kill the cancer cell. If it takes 2 down, we're still in good shape. It'll go after it. And so we just finished writing that study, and we'll start enrolling patients probably sometime latter part of the summer. And I'm already anxious and in anticipation and hopeful that this is going to be another level of game-changing therapy for our patients.
But what I hear you saying is that really your work is never done. And while Katie fell into that 41%, you said that's not good enough.
Absolutely.
We need to keep doing more work so that more people, and that percentage continues to go up.
That's right.
Jenny has a question for you, Katie. What do you make of the irony? Being somebody who's a self-healer, you love to help others, you're a tarot card reader. So what did you make of the irony of coming into this trial and letting science and Dr. McGurk take over?
You know, I haven't really thought about it all that much because I am just so grateful. However, when you and I spoke, when I was at the reading, I thought, oh, wow, this is ironic. But I truly believe that the universe conspires in my favor and that the timing was perfect. Dr. Shanae, you know, the staff, everybody. I have an amazing family. So I really didn't have, you know, any doubts. I was like, let's go. Let's do this.
Erica wants to know, Dr. McGurk, is there a company that owns this therapy the way a pharmaceutical company owns different drugs?
There are multiple companies, hundreds of companies that are working on CAR T-cell therapy, gene therapies in general. For example, for sickle cell disease, in the context of a stem cell transplant, we have a curative therapy these days for sickle cell disease. We've not done a great job of getting the word out among the affected patient populations, but there are companies. CRISPR Therapeutics has a gene therapy for sickle cell disease that appears to be curative in the patients that have been treated so far. But CRISPR Therapeutics is just one, but a leading company in this space.
Last question is from John. Are there any dangers of gene editing? Could you make a mistake in the edit?
Yes.
Does that happen? Tell us about it.
Yes, that can happen. And so this is a really very important question. When the cells are edited, they can cause what are called on-target edits, what are intended to be on target, off of the target in the genes and cause chromosomal breaks. And those chromosomal breaks have the potential to give rise, for example, to a cancer, theoretically. And so there is a very, very intricate screening that goes that's undertaken to look at all the cells and see if there are these chromosomal breaks. Now, with new modifications of the work that those 2 Nobel Prize-winning scientists discovered, the editing is much, much more precise now. So this off-target editing and gene damage of the chromosomes is being avoided with much higher fidelity. But it still remains a theoretical concern. And so again, it's necessary to follow these patients for years afterwards to make sure we don't see something down the line that's going to be problematic. But keep in mind, we're talking about patients who have weeks to months to live. And so one has to look at what are the potential theoretical risks down the line and what are the potential benefits. And I think Katie's alive today in this room because of the potential benefits. there are upfront toxicities we know about for these CAR T-cell therapies, an inflammatory response, the flu times several-fold because the T-cells are attacking just like they would attack the flu, release molecules that cause fevers and chills and low blood pressure, et cetera. And so we know about those, and we have a good handle on how to manage those toxicities. We don't know about 15 years from now. Mm-hmm. The first patients who were first treated in clinical trials are going to come up pretty soon on 15 years, and we haven't so far seen these late effects of other cancers related to the genetic manipulation of these cells. But we have to be mindful and watchful.
So it's this constant balancing act.
Absolutely.
We're going to get to our final takeaways today. Quickly tell the story about when you first met Dr. McGurk.
Well, it was in a Zoom call, and he had a mask on, and he mentioned, "You can't tell I'm really good looking underneath this mask."
Right.
But I'm Dr. McGurk, and I'm part of the head of CRISPR, and so I was like, I like you already.
You're my kind of guy. I said, Dr. McGurk said that? I love it. I knew he had it in him, so he's not serious all the time. So I want to get just final takeaways. Just what do you want folks to know just about your health journey, entering into trials and trusting the science?
Well, I think it's very important to keep an open mind and a positive attitude. That's huge. But trust your gut feeling. I am so grateful for KU and the staff here that they always, nobody made me feel like this was not a good option. They didn't push me, but it's like, this is what it is. So, this is what we're going to do. You sign here, we're going to, the clinical trial, like you said, I'll have, care for 15 years. Mm-hmm. So I know they're going to keep an eye on me. So I think it's very important to get the word out so other people can be, it's amazing. The T cells go in and attack your cancer, and there's no graft versus host. That's amazing. That's amazing.
You have been a great example, and we appreciate it. You've become a friend to us down here and all of the clinicians, and we appreciate you letting us share your story here today. Dr. McGurk, takeaway today. What should it be?
I think the most important message Katie just delivered, and that is people need to educate themselves. They need to be aware. The majority of patients who might benefit by these therapies do not access them for a variety of reasons, and so we've got to get the word out to patients and their families. I gave the example of sickle cell before. The lymphoma patients, only 30% of patients will receive CAR T therapy out of the patients who are potentially curable. And so we have to get the word out to the community. It doesn't do any good to have a very high-tech, highly effective therapy that's potentially curative sitting on the shelf and patients don't even know about it. Mm-hmm. Their families don't know about it. So I think that is that's the message of the day, and there's enormous hope. We're just at the beginning of this extraordinary revolution. I am like a kid in a candy store. It's gee whiz science, as I've said to you in a corny way before, but I believe that. My wife says never say that, but I believe it. Now the other thing is the truth has been revealed that I should have kept the mask on, right?
No, you're maskless in all your glory. So thank you, and good luck in Paris in a couple of weeks presenting all of this information. We appreciate you, and you'll be back to share more.
Thank you, Jess.
Thanks.
Thank you, Kate.
All right, Dr. Hawkinson, wrap us up.
Yeah, as Dr. Stites always likes to say, faith, hope, and science. And I think we've seen two great cases of continuing to have faith here, and then always, as Dr. McGurk talked about, hope and science. We have seen great things with science with the mRNA vaccines, and certainly with this story here today. And things will continue to improve and get better. And so just have to echo Dr. McGurk's final words as well.
Subscribe to our morning medical update and open mics with Dr. Stites podcast. Now everywhere podcasts are available.